introduce Dr. Nancy. Oh, see, I missed it again. I'd like to introduce Dr. Nancy Spinner, Children's Hospital of Philadelphia. We like to call her our Alagille geneticist expert. We're so thrilled to have you here again. Thank you so much for the time you spend with Alagille families and in partnership with us. We just love you so much. Take it away.
Thank you so much. And the love is mutual. And thanks so much for organizing this. Thank you, Stacy. Thank you, Sharon, Roberta. And it's a pleasure to be here. Oh, the first thing, is there a trick to moving the slides because my normal thing is not working? Huh. Let's see. My slides are not moving.
Nancy, you can either use your forward and back button or down at the bottom of your PowerPoint, if you take your cursor down there, the arrows will show up sometimes.
Okay, let's see. Let's move this away. Ah, there we go. Okay, thanks. Thanks, Stacy. It's the arrows on the bottom are working. Okay, great. So hi, everybody. It's a little disconcerting not being able to see the audience and know who's there and know how many old faces versus new faces or scientists versus families, but I will do my best. My goals today are I'm going to talk a little bit about what is genetics genomics, talk about finding the gene for Alagille syndrome, talk a little bit, or the genes for Alagille syndrome, talk a little bit about what we know about jagged 1 and notch 2, and then focus a little bit, or at least talk about one of the big question marks right now, which is how do you explain the variability of expressivity? And that just means that, as you all probably know, some people are very sick and some people not so sick. And why? And we don't, that's an incompletely understood thing right now. So we are going to be talking about, and you can really see it in this symposium, we have the really fortune to work at the intersection of medical science and basic science. And it's really this incredible partnership that you will see throughout this meeting that makes all of this happen. On one hand, there's the clinical or the medical world where you come to see a physician or a nurse or a genetic counselor. And of course, the patients, because none of this happens without the relationships we build with the patients. And that intersects directly with clinical research. So some of those physicians or nurses or counselors, might say, we don't understand this disease. We want to start to think about it. And they start a project to do clinical research. And then that gets coupled with basic scientists, people who work in the lab, who try to understand the basic biology behind disease. And of course, I don't have to even explain this anymore, because I think with COVID, with everything we've witnessed with COVID-19 in the last year, you can really see the importance of things happen. Patients get sick, doctors see the patients, and then you don't understand what's happening. So that's going on all over medicine. And the story of how we worked on Alagille syndrome is really a brilliant example of that. So I'm a geneticist. I have a PhD in genetics. And so let's just think for a minute about what do we mean when we talk about our genetics. And the word we use today is the human genome, which I have to say, our understanding of it didn't really exist when I got my PhD. But we know that each person has 100 trillion cells in their body, and each of them contains a complete copy of our genome, which is all of your DNA, all of the information that goes into making a person or any other organism. The DNA is packed into 23 pairs of chromosomes. Humans have 46 chromosomes, 23 pairs. And the human genome comprises about 25,000 genes. That might be a little high. I think the current estimate's around 22,000, but something like that. 22,000, 25,000 genes that go into making up a human, that allow us to construct our bodies and function in all of the ways that we function. Genes all consist of a specific DNA sequence, so that may seem obvious, but picture these 23 chromosomes that have all of this DNA, 25 codes for 25,000 genes, and it's all a very particular sequence. And these 3 billion base pairs are all tightly wrapped up in every cell. So I'll show you I'll explain a little more about what I mean, but genomics, which is kind of the field that human geneticists today talk about is the scientific study of our whole genome. So in my laboratory, we do research focused on things like Alagille syndrome, but we also diagnose many, many different diseases because I run a clinical diagnostic division as well. And we look at maybe we look at 3,000 or 4,000 of those 22,000 total genes, the ones we understand what diseases they cause. So I always have to start with this picture. Those of you who've seen me talk before know that I like thinking about the history a little bit because it's a relatively recent history. In 1990, the Human Genome Project was launched. And this was launched with the purpose of identifying all the base pairs in the human genome. It was actually initially launched because New Mexico was starting some of the nuclear testing that happens at the Los Alamos lab. And they wanted to be able to track the effects of radiation. So if you want to look at the effects of radiation on your DNA and see if there are any changes, you first have to know what your starting sequence is. And very quickly, the NIH joined. I think it was the Department of Energy that started the project. And then very quickly, the NIH joined in. And it's been a long project. The first draft of the human genome was unveiled in 2001. And at the time, it was a little controversial because a lot of scientists felt like it was kind of a boring endeavor just to brute force go through and sequence the entire genome. But it has turned out to be every bit as exciting as those who were supporting it initially thought. So these were the goals as things started. The argument was that you were going to learn about the structure of the genome. You were going to learn about the biology of the genome. How does the genome work? How do you transcribe genes? Which genes are active? Which genes are silent? How are they organized? And then once you understood the biology of the genome, you would understand the biology of disease. And that would then push you into the science of medicine. And then once you understand those underpinnings, the genetic underpinnings of disease for medicine, then you would be able to make impacts on the effectiveness of healthcare. And I think this has been, this you can see goes to 2020 and beyond. And I think that's exactly what's happened. As many of these aims have been very well, nothing's complete, but many of these aims have been very well accomplished. And today we are still learning to use all of the information we have for the effectiveness of healthcare. That's definitely a big push for the next decade. But the project's been very successful. So thinking back again, we have 2.3 times 10 to the 9th base pairs of DNA. And what you can see here is that DNA molecule that gets packaged into our human chromosomes. I told you that we have 23 pairs of human chromosomes. This is a pictorial representation of a cell chromosomes from one cell from a male. And I know it's a male because women have 2 X chromosomes, and here's an X chromosome and a Y chromosome. And some of the sex determining genes are on that Y chromosome. So you can identify these chromosomes. And this was actually the first genetic test that was available. And we'll come back to how that relates to Alagille syndrome in a minute. But I just wanted to go through some of the basics as we think about the gene, jagged 1 gene and the notch 2 gene that we're interested in here today. So we have DNA, it's packaged into chromosomes. We can look at these chromosomes as a first step towards saying, do you have all of the right genetic information? This is a very gross level. The techniques have gotten better and better and better, and we do many other tests that have much finer, but the Alagille story actually started with chromosomes, so I started there. So what does DNA do? Why do you care about DNA? Why does it matter if you have a change in your DNA? This is called the central dogma of molecular biology, and it just says DNA gets translated into RNA. DNA is a double-stranded molecule. It gets translated into another form, makes a copy that's highly related, gets transformed and gets transcribed into RNA, which gets made into proteins. So proteins are actually the powerhouses of our body. Proteins can be structural. They can they form our organs. They can be enzymes. I'll show you in a minute. but DNA makes RNA makes protein. So DNA is that blueprint. And that's whatever that initial blueprint says, tells you how you're going to go ahead and make those proteins that are actually going to do the work within your body. So a couple of more introductory concepts I just wanted to show you. So we have 3 times 10 to the 9th base pairs of DNA. Only about 1 to 2% of that DNA codes for genes and proteins. So a very small amount, and we're still understanding what all the other non-coding DNA does, but we're learning a lot about genes, and we certainly know a lot, for example, about the jagged 1 gene or the notch 2 gene. And most genes have the basic blueprint is the same. They have what's called a promoter, which is kind of the beginning. That's where the transcription into RNA starts. Genes are made up of regions called exons. These are the parts of that gene that are going to go into making the protein. And then there's this intron. So here you can see you have exon 1, exon 2, exon 3, which are interrupted by introns. Intron 1, intron 2, intron 3. Now, for reasons that are incompletely understood, certainly evolutionarily, all of the introns get spliced out when the RNA gets made. So we're going to end up with a protein that is only directed by the information in these exons. So you can see here that when DNA makes RNA, you splice out these introns, and you have to do that very carefully. And sometimes mutations in those splice sites are responsible for causing diseases. So you splice out all that RNA, all those introns, you make your RNA, and then this RNA molecule is what gets translated into a protein. What do proteins do? As I started to say before, they can be enzymes that catalyze reactions throughout your body, and you heard about some enzymes that are important in the liver from Dr. Piccoli in the last talk. There are structural proteins, things that make up your hair, your nails, your muscle. Every organ in your body has a structure. There are proteins that transport things throughout your body. There are parts of the immune system, your antibodies that we've all been very busily making to the COVID virus in the last year. There are clotting factors that help prevent your blood that prevent you from keeping to bleed, so that your blood can clot. There are other kinds of messengers. There are receptors that are there to receive messages from other molecules. There are parts of your muscles. So there are a lot of things that proteins do. So the whole process of having a very good blueprint so that intermediate RNA gets made properly, and then the proteins get made properly, is kind of what genetics is all about. So when you do a sequence of a gene, it would look like this. You all may remember that there are 4 nucleotides that make up DNA, A, T, C, and G, adenine, thymine, guanine, and cytosine. And so the sequence is just laid out as A, T, C, T, G, C, T, G, C, A. So this wouldn't mean anything to you until you study genes and you understand what this is actually, which parts are the intron parts, which parts are the exon parts, how to make sense out of this. And because so many scientists have studied so much of this, we have a lot of knowledge about many genes, and in fact, most known genes today. So what I'm showing you here, this is a copy of the beta globin gene, which is one of the proteins that's part of your hemoglobin molecule that carries oxygen throughout your body. And so what I'm showing you here are the nucleotides, the ACGTs that are part of the introns, and those that are part of the exon. So here you can see that this is a typical gene. There's a part in the beginning that is just maybe the promoter, where you have enzymes that are going to make a copy of the DNA bind. There's always this methionine right at the beginning of any gene, starts with a methionine that gets clipped off, and then you have the amino acid sequence. The amino acids are a part of the protein that are getting made, but these are the nucleotides that will get turned into protein. Then you have an intron. There are some splice signals, and I'm again just mentioning it because if you have mutations in those splice signals, that's one way to knock out the function of your protein. So here you have part of the RNA that's going to become protein, splice signals, and then this big piece of intron that's just getting lopped off. Then it repeats again. Here you can see you have a really big intron. So it's interesting that oftentimes, like jagged 1, for example, is about 38,000 base pairs long. But if you look at the RNA, it's only about 5,500. So more of the protein is spliced out than is kept. So kind of fascinating evolutionarily when you think about why on earth would your cells carry around all that DNA? It's just going to splice out. So these are questions to be answered. So again, DNA is transcribed into RNA and RNA is translated into proteins. Now, you might also remember from biology that translation occurs via a triplet code. So every 3 bases signify a specific amino acid. So this is the table that tells us what the code is. So if you have DNA uses a thymine, and in RNA it gets changed to a uracil, but you don't have to worry about the details. The point is that every 3 nucleotides specifies 1 amino acid. And you can see sometimes here are two triplets that both code for phenylalanine. And this is just showing you PHE is 1 abbreviation. That's the 3-letter abbreviation. F is the 1-letter abbreviation. And again, I'm only saying this because some of you may see this on reports that you get when it's talking about the effect of a mutation that you might have. So this is just gives you the code. So for any given, so you can see if your screen is big enough that there are underneath each 3 of these base pairs, it's telling us what the normal amino acid is supposed to be. So if you have a mutation, you might have a change in that amino acid, and that might lead you to having a mutation that affects the functioning of the hemoglobin molecule. So this is just, again, a picture showing that you have a DNA sequence. It gets properly spliced down. And then that RNA that's made after splicing has a triplet code that gets read on the protein making machinery, the ribosomes in the cell. And then you make a protein, and here you can see that final product. So I know this is review for many of you, but I just wanted everybody to remember this. So with that background, my involvement with, as I said, I'm a geneticist. I got a PhD working on something else. I was on a basic science campus in Berkeley, California. And then when I came to the Children's Hospital of Philadelphia, I was introduced to this disorder. And again, coming to a place like CHOP was so incredible for me because I had the opportunity to interact with physicians who were seeing patients and presenting clinical problems that we could partner to try to solve. So this is a picture of Daniel Alagille, who was the man the French gastroenterologist who first described this syndrome. And he described a syndrome of liver defects, and I'm just showing you pictures to represent these things, but you've already heard about the liver and the heart from people much more skilled than I am at this. So he described a syndrome that had liver defects, but in combination with very specific other things that formed a syndrome. So as you know, certainly Alagille syndrome is liver defects plus cardiac problems, plus sometimes some skeletal problems. This is a butterfly vertebra that you can see, plus also some eye findings that are not so clinically consequential, but this embryotoxin or this extra line that you can see allows you to have a better diagnosis. And then, of course, the characteristic face, which that's not a characteristic face of Alagille syndrome, but you'll see other pictures of the children. And again, I don't have to tell you, it's fairly recognizable, certainly to everyone who has a child with Alagille syndrome. So this is David Piccoli when we first started. I don't remember what year the picture's taken, but he and I started working together in 1992. And at that time, he introduced me to this disorder that I had never heard of, this rare gastroenterology disease. He had seen a number of patients with the syndrome in his clinic, and he was nobody knew what the gene was, and he recognized that this was a problem that should be solvable in 1992. And again, Human Genome Project launched in 1990. So this was a really wonderful opportunity for me because it was a tractable problem. We had an approach that we thought was going to work. He introduced me to the Alagille syndrome community, which has become a huge part of my life, a very rewarding experience in so many ways. And this was one of the first meetings that we had. This was the Gaymans, Tina and Lowell Gayman, who are here, had a picnic in the area every year. And a lot of folks from CHOP mixed in here are some of the CHOP staff and doctors and nurses, as well as some of the patients. And I'm sure you can pick out various patients who are Alagille syndrome patients just by looking at them. So again, at the beginning in 1992, we knew Alagille syndrome was genetic because it ran in families. And that's kind of the definition of a genetic disease. So this is just showing you it was known to be dominant. And when we say that a disease is dominant, we mean that if you have, we all have, and you saw it on the chromosomes, we have 2 copies of every chromosome. One we got from our mom, one we got from our dad. But if you have 1 copy of the Alagille gene that isn't working quite right, you will have Alagille syndrome. That's called dominant. You only need 1 copy that doesn't work to have the disease. That's in contrast to the other most common, common form of inheritance, which is called recessive. And often we say autosomal dominant or autosomal recessive. And that's because the autosomes are all of the chromosomes except the sex chromosomes, the X and Y. And the X and Y just have a different pattern of inheritance because females have 2 X's, males have 1 X and 1 Y. So it just changes things a little bit. So we speak about them separately. So autosomes are all the other chromosomes, numbers 1 to 22. And there were enough pedigrees showing that there was inheritance from one generation to the next to suggest that Alagille syndrome was in fact a dominant. And this is not an Alagille pedigree, this is just a pedigree illustrating dominant inheritance. So when I started working on this, it was known to be a genetic disease. It was known that it was a dominant disease. And there was a clue because in 1986, it had been published by a different group that there were some patients with Alagille syndrome who had a deletion on chromosome 20. Might be a little hard to appreciate, but this chromosome here on the right's a little bit shorter up in the P arm. So this was exciting because there were several patients who had deletions on chromosome 20. And this told us, oh, the gene is on chromosome 20. So now we knew where to start looking. This is Ian Krantz, who was a fellow at the time and worked on identifying the gene. This is Liz Rand, who's a gastroenterologist at CHOP, who had moved from the University of Chicago and had already started collecting Alagille syndrome patients because she was interested in trying to identify the gene as well. And she came to CHOP and we worked on these patients together. So we had collected the first series of, I think we had 25 or 30 patients. It was not an enormous number. This is family 1 for us. And this is a family with 2 little boys who both had Alagille syndrome. And mom had some mild features. She actually had what we now recognize as the adult face, but we didn't really know that. And she had some mild liver abnormalities, although she didn't even pay attention to them because she wasn't clinically sick. She had some mild skeletal findings. But again, she really, she wasn't thinking about this, but then when they had 2 children with Alagille syndrome, it became something that she thought about. They were the first cohort of patients that we started studying. And in the early days, we used to, instead of just sequencing things, we ran these gels. I won't bore you with the details, but this is actually, you take DNA, you put it into, it's like a large jello that's placed in a mold and you apply electric current. And under the right conditions, you see how the DNA migrates through that large gel. And this is what we're looking at here. And if we had 100 other normal individuals without Alagille syndrome, they would all look like these individuals. So these 3 that are at a different, are running at a different spot in the gel, this was telling us that there's something that's going on here. So once we saw that, we don't do this technique anymore. We just go right to sequencing, but historically it was kind of interesting. So once we saw that, then we would go and sequence, and this is 1 exon, jagged one has 26 exons. This was exon 4. We went back, we sequenced that exon, and sure enough, interpretation of this sequence tells us that there's a 2 base pair deletion. So we now could see that, yes, in fact, jagged 1, which is located on chromosome 20, right in the region we predicted was going to be the critical region for this, there is in fact a deletion for this patient. So what happens when you have that deletion? And remember, we went through the triplet code. So now you've taken out 2 base pairs. So geneticists, people teaching genetics sometimes use the example of if you had a sentence that was composed all of 3-letter words, you know, the cat sat hat, and you took out the H and the E from the and still read it in groups of 3, now the words make no sense. So that's kind of what happens here. You take out those 2 base pairs, everything downstream gets shifted. And in this case, what happens is that out of all of those codons, those triplets that code for the amino acids, there are 3 that code for a stop signal that say stop here, because you have to know when to end the protein. And so in this case, the protein stops way too early. And so this protein can't do anything it's supposed to do. Jagged 1 is supposed to sit in the membrane of a cell. This protein, just doesn't have the transmembrane domain, and it will not function at all. So this is a really good example of a JAG1 mutation that clearly is going to lead to a non-functional protein. So we were able to identify JAG1 as the disease gene really because of its location. And again, I'm sparing you all the details, but it took us 5 years to map the region and figure out the genes that were in the region. Some of the excitement did get kind of exciting, and Alagille, the movie, you can go and see that. But what was JAG1? Like why this gene? Turns out JAG1 was a member of the Notch signaling family. There wasn't really that much known about the Notch signaling family in humans at the time. And we published this discovery in 1997, but it was known as an important signaling pathway in the fly. So here's a normal fly wing. Here's a fly with a notch taken out of its wing. And this is how the pathway was first studied. So it's a basic pathway that's involved in the development of many different structures. Nobody could have guessed that JAG1 would have been the disease gene for Alagille syndrome. So it was part of the excitement for us. And actually, there is no JAG1 in the fly. There are several other Notch ligands. And what you can see here is the way JAG functions is that JAG1 is what we call a cell surface ligand. So it's a protein that sits on the surface of a cell, and its job is to find other cells that have the Notch receptor on the cell. And when a JAG expressing cell and a Notch expressing cell come in contact with each other, things happen. So that relationship spawns a piece of the Notch receptor called the intracellular domain actually gets decoupled from the part that's on the outside of the cell. It moves into the cell where it binds with some other proteins and causes gene transcription, and that actually affects cell fate determination. So differentiation. So how do cells decide what they're going to turn into as an embryo is forming? There are many, many places where you start from a small undifferentiated bundle of cells. Now you have to make a liver. So there are so many coordinated things that have to happen and all sorts of signals. One of them is the Notch signaling pathway. And I think you'll hear more details about the science of that tomorrow. So here we had this cell fate determination pathway. We proved that JAG1 mutations caused Alagille syndrome. And we spent the next few years furiously screening patients and trying to look at the mutations and understand more about it. And then we were finding mutations in about 60% of patients. And then in 2002, Tom Gridley's group published a paper showing that there was in the mouse, if you had a mutation in JAG1, it wasn't quite enough to give you Alagille syndrome. But if you had a mutation in JAG1 and Notch2, you could get Alagille syndrome. So this led us to start to screen the Notch2 gene. And sure enough, we identified some families that have mutations in Notch2 that cause Alagille syndrome. And there are many fewer of these families. At first, it looked like they might have more kidney renal defects, but as time has passed, that hasn't really panned out. So there are a few differences. Patients with Notch2 mutations do seem to have a more variable phenotype. The facial features aren't usually quite as striking, so it can be a little harder to diagnose. There are a couple of probands that had non-syndromic bile duct paucity, so no facial features, no cardiac defects. So we don't understand this quite as well. And there are so many fewer mutations. And we make maps of the proteins and try to understand where are the things that are going to be important. But I would really say this is a work in progress. And I do think that Stacey Huppert is going to be addressing some of this tomorrow, because it sounds like her talk is focused on Notch, science overview, and Notch2. So that will be great to hear more about what's happening there. So where are we today? So in our laboratory, we have studied over 400 patients at this point. And this is just a diagram showing you that here are the mutations in JAG1 spread out across the top. These are all of the different changes that can be found in the general population. So one of the things that gets complicated about genetics is that we all have mutations. We've got lots of mutations, but many of them aren't disease causing. They're just DNA changes that make us who we are, right? So they're called polymorphisms, things that come in different forms. And you probably can guess that, and probably makes sense to everyone, because we all have different heights, different hair color, different eye color. There are lots of things that go into making us who we are. But some changes are disease causing. So up here on the top are the JAG1 mutations that are disease causing. And you can just see there are lots of them. There are lots of different mutations, and they're mostly spread out all over the gene, pretty much solely restricted to the part of the protein that sits outside of the cell. This is the transmembrane domain, and this is the part that's intracellular, that goes into the spaces inwards. And we haven't seen any mutations that we can be sure of there. And you can see that for Notch2, there are just a lot fewer. So this is still a work in progress. I don't know that the types of mutations are a little bit different. And I would say we understand a lot more about JAG1 compared to Notch2. So one of the things that I think is worth mentioning, because for those of you who have had genetic testing, you may have gotten some of the mutations. Any of us in this field can look at them and say, definitely disease causing. We know what that mutation's doing. Yes, that is the cause for your Alagille syndrome. But one type of mutation that occurs in about, I think it's about 11% of patients, is called a missense mutation. So the mutation I showed you in our family one before had 2 base pairs missing. That's called a truncating mutation, because it truncates the protein, and we know that protein's not going to work. Missense mutations, on the other hand, are when a single amino acid is changed, and it's harder to figure out what that's doing, because the whole protein gets made. It's just one spot is different. So some of those are fine. They're just variants. They're normal variants. Some of those are disease causing. And we have studied this and are actually gearing up to do some more work on this, because it's a problem for diagnostics. You're not sure if this is disease causing or not. So I'm sorry, it's 15% of variants in JAG1 are missense. The numbers go up and down a little bit. And every time you see one, you really have to think hard about it. And I do want to make the point that sometimes you get a report from a diagnostic lab that says you have a change in this gene. We can see the change, but we're not sure what it's doing. And they call it a variant of uncertain significance. And that can be very frustrating for families. And there is a definite skill and art to correctly diagnosing these. So this is an example. This was a 2001 paper from our lab where we were studying some of these missense mutations. And in this case, here are 2 cells. This is a normal jagged one, and it's being displayed along the outside of the cell, just like I said, that its job is to sit on the outside and talk to a Notch2 presenting cell. And it should be displayed all along the membrane. And that's a normal JAG1. These are 2 examples of 2 of our mutants, where for one reason or another, it's a missense mutation, but they don't get out to the cell surface. They're getting stuck within the, close to the nucleus, so they're not on the cell surface. So it could be a non-functional one or a functional one, but if it's not getting to the cell surface, it can't do its job. And then we have some other assays where we can look at, is it signaling normally? So each of these can be complicated, and we get a lot of emails from people who are who've received a report, or get emails from physicians who have a patient and they want our opinion, because we don't know that much about everything, but we know a lot about Alagille syndrome and JAG1 mutations. But there is a specific series of steps that one has to go through. And then this is the kind of the guide to how to understand if missense variants are pathogenic. And there's some things we have to look at. So you look at this functional data, like I was just telling you. Does it get where it's supposed to go? Does it signal? Do you see it in multiple unrelated individuals? Do you see it in patients with Alagille syndrome? Is it well known? Does the patient who have it, do you believe they have Alagille syndrome? Good phenotyping means, did somebody actually confirm this patient has the clinical signs of Alagille syndrome? Is it in their family? So there's a whole series of questions that we might ask. We might, if we don't have too much information, we might ask, what about, is the amino acid likely to cause a change? Where is it located? And then there are a number of incredible databases that give us the frequency of genetic changes in the normal population, just a wide swath of individuals. So we can look in those databases, and if it's present at high frequency in a normal population database, it's probably not causing Alagille syndrome. So I just this morning came upon a paper where they were publishing that there was a patient who had two different disorders, and they were both could account for the tetralogy of Fallot and the butterfly vertebra in the patient. And I thought, oh, that's interesting. We usually don't see that happening where you have both a 22q deletion and Alagille syndrome. So I downloaded the paper, took a look at it, and it is absolutely a polymorphism. So even though that paper is in the medical literature saying that this patient has an Alagille causing mutation, they do not. We have functional data that says that it is a polymorphism. It is not a disease-causing mutation. So it's a little tricky, and we're always happy to get requests from physicians or from patients if you have a missense mutation and somebody's struggling to try to understand it. And again, it's not that we're smarter, although I don't know, maybe we are, but I'm kidding. We're not. We just have a lot of experience. And this is the one gene out of the 22,000 that we can say a lot of things about. So as we started screening all of our patients, we found first we had 60% of our patients had mutations. Then as techniques got better and sequencing got better, and we went up to 70%, and then it was 80%. And as of 2 years ago, we were left with a pretty small group of patients who we couldn't find mutations in. There were like 16 of them. Now, we also, every single patient we had in that database, we had David Piccoli and Kathy Looms and Benita Kamath look at them very, very carefully to ask, do they have Alagille syndrome? So that's one key, is that you have to really understand what your clinical diagnosis is. So when one of the experts, or anybody who's highly trained says, yes, this patient meets the clinical criteria, we were finding about 94% of our patients had mutations. So that's pretty good. We had the ones that got away, of course, bothered us. So we asked, why were those, why were we missing those? And Ramki Rajagopalan is an outstanding bioinformaticist who has been working with us for a number of years, just joined our faculty at CHOP as of July 1st, very exciting. And Ramki did a couple of things. As techniques were getting better and better, we went from just sequencing the JAGA gene to sequencing the entire genome, or the entire, you could sequence the collection of exons. We call it exome sequencing. So everything where where you're putting your money because you think that that's where the mutations are going to be, because it's the protein coding part, or the entire genome. So we've now done both things. And as you might have predicted, and as we predicted on the last slide, in a couple of cases, we found it was the wrong diagnosis. So this was part of our, screening through some of these patients. And I think those patients were on the we're not sure list. I know they were. So 2 out of 8 had come in as Alagille syndrome, but we were able to show they had something else. One was a PFIC, and one had a NEC8 mutation, this renal hepatic pancreatic dysplasia, which had been recently reported in a very small number of patients. So we published this patient. So that was kind of exciting to add to that story. And then when he did genome sequencing, which is a technique that's kind of the most sophisticated tool that we have now, we found 4 mutations that had been missed by the other technologies. And I don't have time to go through the reasons for that, but just suffice it to say that the better your tools, the more things you're going to find. And this was really cool because all of these things made sense. None of the other tools that we had used before would have picked these up. We understand that. So we're hoping that genome sequencing will be the predominant tool used for clinical diagnosis. So we're doing pretty good with Alagille syndrome, but of course that hasn't necessarily cured things or given us a complete picture of why jagged 1 mutations cause this disorder. I just want to say a word about haploinsufficiency. So I told you that Alagille syndrome is a dominant disorder. So in this case, just having 1 gene that doesn't work is what's giving you the phenotype. So and haploinsufficiency is defined as the intolerance of a gene to the loss of 1 allele. The allele is just the version on your 2 chromosomes. You have 1 allele on 1 chromosome, 1 on the other. The allele is just the version of the gene that is present. So intolerance of a gene to the loss of 1 allele in an organism that is diploid. So organisms we as humans are diploid, which means we have 2 copies of every chromosome. There are some organisms that only have 1 copy of the chromosome, or some can have more, but most mammals and certainly all humans, we're diploid. But if you're missing 1 of certain genes, you express the clinical phenotype. More human disorders are caused by recessive diseases, where having 1 protects you. So you might have, for folks with cystic fibrosis, for example, if you have one copy of the cystic fibrosis transmembrane receptor that doesn't work, you don't have cystic fibrosis for the most part. You need to have 2 that don't work, or many different metabolic disorders. 1 is okay. In Alagille syndrome, it's not. And it's it's not the only dominant disorder, and I don't remember the numbers, what % are dominant versus recessive, but it is the minority. And haploinsufficiency is a really interesting phenotype, and you might ask why is not having 1 enough? And these seem to be genes where the dosage is very, very tightly controlled. Evolutionarily, you don't seem to want to have too much or too little. And because too much can be very bad, that somehow it gets into this very, very tight range, so that when you lose 1, you have the phenotype. So it's a form of dominant inheritance. It's less common than recessive inheritance, where you need 2 non-working copies to show an effect. So things for us to think about as we try to understand this disorder and possibly how to treat this disorder. So Alagille syndrome cohort from 1992 till 2021, we have 406 probands. Those are the ones that meet clinical criteria. We're now up to 97.5% of patients. So that's pretty good. Hopefully we'll find the rest in the next couple of years. Most of those are JAG1 mutations, 95%, small percent, 2.5 are notch 2. About half of the time, I told you this is a genetic disease, but half of the time it's newly occurring. So neither parent has it, but yet a child is born who has it. And that's because there's a new mutation. And that's another whole part of genetics. We all start from the union of an egg and a sperm, and then those egg and sperm have to there are many rounds of replication that go into forming those egg and those sperm. And then when they get together and start to form that zygote that grows into a fetus and into a child, there are there are trillions of cell divisions. So all along the way, there are opportunities for mutations to happen, because it has to happen with such precision. And mutations are just a part of life. We all have them. We just can have them in a lot of different genes. We can have them in things that don't do anything. One of my favorite papers from about 2011 was they did sequencing of a woman who was 115 years old. And she had, 450 known mutations that were identified that just happened along her lifespan, but none of them did anything that killed her. And in fact, she was so long lived. So that it's just something that happens, and that's what we find empirically. About half of the time, it's a new mutation, and half of the time it's inherited from a parent who might have some features or who might be more mildly affected. So just a few words, we're getting close to the end, on variable expressivity. This has kind of haunted us since we started working on this. We know that there's mild Alagille syndrome and there's severe Alagille syndrome, and we don't know why. And this is one of our Philadelphia families where you can see mom and two of her sons are affected, and they're affected very differently. So one son was quite sick. You can see that he's short, he's quite jaundiced. He had a liver transplant. His brother fared much better, and mom also had a much milder presentation. So why? So being geneticists, we wanted to test the hypothesis, are there genetic modifiers that can contribute to this variable expressivity? That's what we call these different phenotypes. So we did a standard type of study called a genome-wide association study. And we looked at many markers all across the genomes. We looked at our patients and we divided them into those with mild disease and severe disease. That was not easy. The gastroenterologists, and here you can see Benita Kamat, David Piccoli, and Kathy Looms, had to actually work very hard to come up with the right criteria and figure out how to bin those groups of patients, but they did. And then we mapped markers all over their genomes, and we asked, is there anything that's different? And we did come up with something that was different, much to our surprise, which we're still trying to understand. And this was the work of, I don't know what, this must have been in about 2014, I think. But this is Marcella Devoto, who's our statistical genetics colleague, who just moved to run an institute in Sardinia, where she is from. So we miss her from CHOP. And Ellen Tsai, who got her PhD working on this project. And Ellen has gone on to work in industry in Boston and just had her first baby. So it's one of the pleasures of doing this work over a sustained period, is we get to follow people as they go through life, which is really an incredible privilege and really exciting. So we found this gene called thrombospondin 2. We knew nothing about it, but it turns out that it was expressed in the right places. It is known to, we actually collaborated with Michael Wong at the University of Michigan, who had shown that thrombospondin 2 inhibits JAG1 Notch2 signaling, and some other experiments that all said it's kind of in the right place at the right time, and maybe it will have something to do with it. We came up with a model for this, and in this case, we're just showing that the proposed model would be that normally JAG1 and Notch2 interact to stimulate bile duct morphogenesis, the formation of normal bile ducts. Thrombospondin 2 is a protein that can interfere with jagged notch signaling, but it may have a physiological role where it's supposed to do that to some extent. And in a normal person, there's a good balance, and you make all those nice ducts and ductules, as you saw in Dr. Piccoli's talk just before. But if you have a JAG1, one JAG1 doesn't work, then you have fewer JAG1 Notch2 interactions. And we know that in those cases, you have some decreased bile duct morphogenesis. And so perhaps if you have too much thrombospondin 2, which is our working hypothesis, then you make even fewer bile ducts. So this is an ongoing area of study. We have a medical student who's been working on this. That's James Nassar on the bottom. Melissa Gilbert has been leading this project in the lab. Ben Wilkins is a pathologist who also works in the zebrafish, who's been helping. And this is James, who is a Penn undergrad and is now a medical student and is working on this over the summer. He got a NASPGHAN grant to follow up on this. And they're working with Christoph Seiler as the head of our zebrafish core. And so they're designing assays to use zebrafish to look at overexpression and expression of thrombospondin 2 in the context of JAG1 fish that have, a deficient JAG1, to see if we can replicate this. Now, I just have to say, I'm not going to talk about it, but you might hear about it, that there is certainly other work that's going on and other potential modifiers. And I could not give a talk happily without showing one of Stacy Huppert's incredibly beautiful castings, where when she looks at mice that have mutations along the pathway that might be affected, might be related to Alagille syndrome, she's able to show with this beautiful work that you in fact get fewer bile ducts. And this is from work that Stacy did with Hamid Jafar Nejad, showing that SOX9 is another potential modifier. So there's a lot going on. I hope we've made some important observations over the last 20 years that we've been doing this. We've certainly been able to diagnose Alagille syndrome. Having a molecular marker has taught us more about the Notch pathway, and we understand it's been able to be studied in many organisms. We've been able to expand the clinical features of Alagille syndrome, some of the vascular manifestations, which you've heard about, some of the renal manifestations. We didn't know that much about them until we had a molecular marker. We're looking at variable expressivity, and this, of course, does give the potential for treatment. And we are collaborating with some of the groups and the companies that are working on this. It's not a direct area at this time in our laboratory, but we're kind of thrilled if our work is able to help other people to come up with ideas for how you might treat that. And there are a number of ideas that are out there, and I think there's a lot of reason to be optimistic that something good is going to happen. So we view genomics as sitting in the middle of the world of, world of medicine, where we're involved with patient care, with research, with diagnostics, hopefully with treatment, and certainly managing genomic data is becoming big business because there's just so much information and gives you so many clues for other things, related things, modifiers. It's there if we can figure out how to use it. So the future is bright, and I sincerely believe that. I think you'll hear about that tomorrow. This has been an incredible period for me and project and working with incredible people. These are people I currently work with and people who have either left my lab or people who were collaborators, but you guys all know that because you're meeting a lot of them today and you get the idea for what kind of community this is. So I just want to tell Stacy and Kathy that apologies, I cannot come tomorrow. And I just wanted to mention that, as many of you know, my husband's also a medical geneticist. And in fact, he's worked on Alagille syndrome with us for many years. His name's Ian Krantz. And Ian has started a 501c3 charity that supports, well, his way of looking at it was that his goal in life was to merge his love of cars, which is a passion for him, with treatments and help for kids with birth defects. So he has a birth defect center at CHOP in genetics where he sees patients with a wide range of different things. So he started a car show, and it's the 4th annual Philadelphia Concours d'Elegance. And it is an exciting event. If anyone's in Philadelphia and ever wants to come, unfortunately, the car show's tomorrow. So I hope next year the symposium doesn't conflict, and I'm going to make sure it doesn't. But anyway, I'm so sorry I can't be there tomorrow. But thank you so much for your attention. And I think we have a few minutes for questions, which I'm more than happy to answer.
All right, thank you so much, Nancy. We do have a few questions. If there are questions that we don't get to before this session ends, then we'll follow up in the next day or two. First question comes from Amanda. Both my sister and I were diagnosed with Alagille syndrome. Parents were both tested in the 90s and both were negative. How is that possible, and should they get tested again?
Okay, so that's a great genetics question. It is a phenomenon potentially called germline mosaicism. So that can happen where one of your parents may have had a mutation that occurred after the formation, after they started developing. So in their blood, they don't have a mutation, but someplace along the way or within the cells that went to form their eggs or sperm, a mutation happened. So they have some cells with those mutations that are either in their egg or their sperm, and that's why they have a recurrence risk. So if you are a germline mosaicism, a germline mosaic, then you might have to, if you wanted to know whether or not a future pregnancy had Alagille syndrome, you would have to do prenatal testing because it's hard to tell that. There are some places that can do sperm testing because it's a sample you can get, you know, eggs, nobody died on, I'm not aware of anybody who does that. And in a way, it's just a little bit of luck. Are you going to, you know, as to whether or not, you don't know what percentage of your eggs or sperm are affected. So really the only thing to do is to try to do either pre-implantation genetic diagnosis or or take your chances or do regular prenatal diagnosis. The other possibility, of course, is always that there was an error in the lab. So something to think about that does happen very occasionally, but germline mosaicism is a pretty well-known phenomenon.
Okay. Another question, my daughter has never had a genetic test. She was diagnosed via biopsy and clinical symptoms. She is adopted, so no risk of other kids. Is there a reason we should pursue genetic testing for her? And if so, what tests do you recommend?
Well, in today's world, getting the JAG1 gene sequenced is kind of straightforward. So, I mean, I think that's what you'd have to do because you wouldn't know where to look. I would say that it might be useful for her for future if she wanted to know if what the mutation is that she carries. And again, if she clear, it probably won't matter to her medically if you're sure she has Alagille syndrome. I'm only sure she has Alagille syndrome when one of our experts has made that diagnosis, but perhaps there's a wide range of experts across the country. So if you're sure of that, it probably won't matter to her, but it might matter to her when she wants to go and have children herself.
Okay. All right, and then the last question on the chat. This is from one of the families in our Brazilian group. Do you recommend a genetic test for a family, the ALGS mom and dad have no symptoms, but before they have more children?
So if you have a child with Alagille syndrome and you've confirmed a mutation in the child, and if you want to have other children and you want to know if they're going to be affected, then for sure, I think that it's a straightforward test. Once you have the mutation in the family, you don't have to screen the whole gene. So you can just go and have that single exon looked at, which is a simpler, faster, more straightforward test. And then, you would have that answer, for yourselves for the future. It's, it's going to be a personal preference as to whether or not what you would do with that information with regards to future pregnancies. But if you want it, it's pretty straightforward to get it.
Okay, thank you. Let's see. Oh, Philip has a question. Go ahead, Philip.
Yeah, hi. I was wondering, are there any efforts underway to look at epigenetic marks and how they might contribute to the severity of Alagille syndrome? So
that is a very big area right now. And we've actually had some thoughts of looking at that, so for those of you who don't know, the sequence is one thing. And, we try to learn what the sequence is and how that might affect expression. But there are other ways to turn on or off a gene. And that's by marking it with various types of chemical marks that happen naturally. So one of them, for example, is methylation, where there's if a gene gets methylated, it might turn the gene off and gene expression might change. So I'm not sure, there is certainly discussion of that. I'm not personally sure who is looking at that in a systematic way, but there are others at this conference who may know that. So this may come up tomorrow.
Awesome, thank you.
Yeah. But it's a good, it's a good idea and it's an important idea, and it's definitely one of the ways that you can change things.
All right. Thank you so much, Nancy, again, for your time. And we will miss you tomorrow, but all of our best to Ian and his event for sure. We are going to take a short break. We will be back here at 4:30. Again, you'll want to X out of this session, go back into the auditorium to access the next session. While you're on break though, please feel free to go into the virtual expo, take a look at the booths that are in there, say hello to some of our sponsors, our supporters, that have a booth, and then also the lounges where you can connect with each other through chat or Zoom. So thank you again, Nancy, and thank you everyone for joining us. We will see you all back here at 4:30.
Thank you. Bye.
Thank you. Bye.